be available in a couple of days on the HMSC past seminar page. And so if you are interested in seeing this again or sharing it, in the chat box, I just put that link so you can see where that recording will go. And let me see, catching myself back up because I forgot to record. But just a couple of announcements before we get started with today's talk. I wanted to let everybody know that next week, already April 1st, we have Ryan Moeller, who's going to be joining us from the Department of Microbiology at Oregon State University, talking about phytoplankton and the marine microbial loop. So it'll be interesting to hear him kind of bring those two ideas together. So I'm interested in learning a little bit more about that. If you're interested about any of our talks that are coming up, you can just Google or use whatever platform you use to put in HMSC, scroll to the bottom of our landing page, and there's a calendar of events there. But I'm going to stop talking and hand this off to Lee. Lee is the one who invited our speaker today. And so Lee, if you want to take it away, I'll drop off the screen. Thanks, everyone.
Great. Thanks, Cinnamon. Yeah, hi, everybody. Yeah, it's really my pleasure to introduce Solène Derville to you all today. She's my postdoc, but you might notice that she's dressed in a little bit more summer attire than us, and that's because she is actually based in New Caledonia due to the pandemic. She has been unable to join us here in Oregon, even though she started her postdoc with me, I think, in September. So we've been doing the remote work thing, but it's been wonderful. But she might look familiar to some of you anyway, because throughout her master's degree and her PhD, she often spent time here at Hatfield with us, 3-month stints here and there, where she worked with us in the GEMM lab during her master's on Maui's dolphin distribution, and then for her PhD she studied humpback whale distribution patterns around the South Pacific. So Solène recently received her PhD from the Sorbonne University in Paris, but she's been based in New Caledonia for a long time. She's interested in animal behavior and spatial ecology, basically the way that animals interact with their environment and move and how they're distributed in space. So she's really a stellar rock star spatial statistician, and she often develops innovative and multidisciplinary ways to understand the scale at which animals, particularly marine megafauna, use their environment. So I was super excited when she joined us on this project that she's going to be telling you about now. That's really important to many of us here in Oregon. It's, involves our fisheries and our communities and our whales. So we're really grateful to have her part of our project. So with that, Solène, you can take it away.
Yay. Okay, thanks, Cinnamon. Thanks, Lee, for the introduction. Hi, everyone. I'm very happy to be presenting my work and the work of the GEMM lab for you today. So I'll be presenting the methods and preliminary results that we obtained from combining aerial and boat-based surveys to model large whale habitat year-round along the Oregon coast. Okay, so first, a few words about the context of this project. As many of you know, reports of whale entanglements have been on the rise since 2014, and at a rate of more than 20 entangled whale every year over the US West Coast. However, this number is likely to be an underestimate of the true entanglement rates. So when gear could be identified, Dungeness crab gear has been most commonly found to be the cause of entanglements. These entanglements are mostly impacting humpback whales of two different distinct population segments, breeding in Mexico and Central America, and respectively listed as threatened and endangered in the Endangered Species Act. Now, humpback whales migrating and feeding along the coast of Oregon could also include a small proportion of the Hawaiian DPS, which is not listed under the Endangered Species Act. Blue whales of the eastern North Pacific endangered stock are also of concern here, as well as gray whales, particularly the small Pacific Coast feeding group that feeds off the Pacific Northwest during summer and fall. The Oregon Whale Entanglement Working Group was formed in 2017 to address this issue of whale entanglements. And the group identified the lack of understanding of whale spatial distribution in Oregon as a limiting factor to take effective and informed management decisions. So here is where the OPAL project comes in. OPAL stands for Overlap Predictions About Large Whales. And the objectives of this project are to better understand the environmental drivers of whale distribution in Oregon waters and how these patterns of distribution change with season or year. And from these models to be able to predict whale distribution and anticipate the co-occurrence with crab gear. So here is my framework. So I'm not in the field to collect the data, but I'm doing the data analysis part of this project. So first I'll show how I compiled cetacean distance sampling survey data, looking both at survey efforts and baleen whale observations. Then how to model whale occurrence with respect to environment using the standardized whale counts as a function of static and dynamic environmental variables. And finally, how to predict whale dynamic distribution. So how to extrapolate predictions from those little transects that were surveyed to the entire Oregon coastal waters, seasonally as well as daily. So the OPAL project is mainly based on an amazing collaboration with the United States Coast Guards that have allowed us to fly in helicopters with them on the Oregon coast. So here, Craig Hayslip and Lee Torres are the people who have been running all of these surveys on the Oregon coasts. The helicopter flights are conducted from 3 different stations. One is from Astoria. It's called the Warrenton Transect. Then there's another transect leaving from Newport up to Pacific City. And then from the North Bend station, we have 2 different transects, the NB North and NB South. So you'll see those 4 transect names several times throughout this presentation. Now, all of these flights are conducted at 500 feet altitude and 90 knot speed. There's 1 observer aboard, and they have been repeated on a monthly basis since February 2019. So the OPAL project could have gone entirely with these helicopter surveys, but we also thought that this was a good opportunity to use our boat-based data. Because the GEMM lab has been placing marine mammal observers on the Shimada on the Northern California current surveys since 2018. These observers record marine mammal observations during transit between stations. Here, this map shows the NCC stations from southern Washington State to Northern California and going offshore up to 200 nautical miles. And in addition to these surveys, the GEMM lab has also been conducted STEM cruises. So the objective of these cruises is to take students out on the water and show them what an oceanographic survey is like. And in addition to being great outreach and great experience for those kids, it's also a good way to collect data. So we've also been using that distance sampling data from the Oceanus. So all of these surveys are conducted at 8 to 11 knot speeds. There's usually 1 to 2 observers at the same time, observing at a height of about 10 meters, which is really not too bad to be able to detect whales pretty far away. And these surveys have been taking place in February, May, and September since 2018, with also an additional Oceania survey in 2016, which I have included here. Now, in the field, whether working from a helicopter or from a boat, the same distance sampling protocol has been applied. Transects are surveyed with naked eye and binoculars, and any change in conditions that might affect detection is recorded. Upon detection, the distance to marine mammals is estimated with 3 different methods depending on the platform. So from boats, we use visual estimations of distance as well as binocular reticules. So this is what Don here is doing with a student on the Oceanus. And from helicopters, we've been using visual estimations as well as geometer. So a geometer is that orange box that Lee is holding in front of her eye on the photo. And it measures the vertical angle between the observer's eye, so the observer being in the helicopter, and the target located at the surface of the water, which is the whale. And distance to the target is calculated from the roll angle using simple trigonometric calculation. Now, why do we measure distance and what do we do with it? Distance sampling relies on the assumption that while surveying a track line for cetaceans, a proportion of animals present in the area are missed, and this proportion increases with increasing distance from the track line. So the probability of detecting a cetacean in the vicinity of the track line is modeled as a detection function, which I represented on this plot. So the detection function is Gx, and x is the distance. And as you can see, the slope is decreasing here. On the track line, the detection is assumed to be absolute, meaning that all cetaceans surfacing on the track line are expected to be detected and counted. So in practice, this means that Gx, when x equals 0, Gx equals 1. So the curve starts here at 1. Now, a very important parameter that I will be deriving from this function is the estimated strip width. So this is the distance at which as many whales are seen beyond it as are missed within it. So that yellow area here on the plot represents the whales that are missed, and we're trying to place the red line, the estimated strip width, at a distance that would allow the blue area to be equal to the yellow area. So in the end, the estimated strip width allows the estimation of the area that is effectively covered during the survey. And this is the parameter that I will include in my model later on in the analysis pipeline. So another important aspect of the distance sampling protocol is to keep systematic records of any change in condition that might affect detection. So we're recording glare, weather, buffer sea state, visibility, and so on, but also parameters that are related to the observers or the platform of observation itself, such as the number of observers, the height of the platform, the speed, and so on. So here, because we are actually combining different platforms of observation together, these differences in detection will be very important to account for survey efforts. So to standardize the spatial analysis of our cetacean sightings related to environmental variables, the survey effort must be segmented into equal distances. So to do so, I split the track line into segments of 1, 2, 5, or 10 kilometers. This is what I represented here as an example. So here is a helicopter survey conducted from North Bend. As you can see, a part of it is missing. This is because the conditions here were not good enough to observe. So maybe some fog or rain. And this is the kind of section of the track line that I would be removing prior to the analysis. And then segmenting this track line into little segments of 1 kilometer, 2 kilometers, 5, or 10. The optimal segment length was selected among these 4 options based on a trade-off between limiting the number of segments with 0 detections and maintaining a high spatial resolution. And so far, I found that the 5 kilometer segments, so the one here in the middle, gave the best results. And so I've been using that segmentation for the rest of my analysis. Environmental layers were compiled for the study area, and environmental predictors were extracted at the center of each of these 5 kilometer segments. So as prey distribution data are often lacking in our habitat models, the whale ecologists often resort to using environmental proxies. So variables such as topography or ocean circulation have proved to be good predictors for fish or krill distribution, and in turn, potential predictors for whale-suitable habitats. So here I have acquired and mapped depth, distance to shore, slope, aspects, all of these to describe the topography of the region at 500-meter resolution. The dynamic predictors are the ones that change for time, and they can be acquired from remotely sensed sources. So those are the satellite-derived variables, and from regional ocean modeling systems, so the famous ROMs that have been used on several occasions to predict predator distribution in the California current system. So here my satellite data includes temperature, sea surface temperature, sea surface height, eddy kinetic energy, and chlorophyll A. I'm also deriving standard deviation from those, so that's the SD, because this is an estimate of the spatial variability of these parameters, and it may be used to detect frontal zones, which we know are important to predators. The ROMS variables include surface parameters, but also some subsurface parameters, the isothermal layer depth and the bulk buoyancy frequency. So specifically, the isothermal layer depth, the ILD, is a proxy for the mixed layer depth, and it indicates the depth where physical water properties such as temperature, salinity, nutrients, oxygen will change dramatically. I represented the spatial resolutions here, and as you can see, they vary quite a bit from one source to the other, from 500 meter to 20 kilometer. And temporal resolutions are also something that can vary, and I've been testing different things. So for those dynamic variables, I've been averaging them on a monthly basis, or looking at them at a daily real-time scale, or at a daily scale, but with an 8-day lag prior to the survey. Now, I'm not going to describe those variables into too much detail, but I just meant to show you how these can vary both spatially and temporally. So this is an example of several variables from June 2019 to April 2020. So at the top, I represented the sea surface temperature, sea surface height in the middle, and isothermal layer depth at the bottom. And as you can see, there's quite a bit of variation, longitudinal variation and seasonal variation going on. So we'll see how this might affect our well distribution. Now, once we have the whale data and the environmental data, it is time to produce our models. So this model will relate the number of individuals detected with environmental variables and temporal variables, and it should be able to account for the area that's effectively surveyed per segment, the spatial temporal autocorrelation in the data, and its hierarchical structure, because the data is structured by year and by platform. Finally, the study objective is also an important thing to consider to decide on which modeling approach to use. So descriptive models tend to reveal the general patterns of habitat selection with respect to each environmental variable, while predictive models fit more closely with the data, but might have a lesser performance when predicting distribution in unsampled areas, unsampled times or conditions. So given the OPAL objective to accurately predict fine-scale patterns of well distribution in Oregon, I chose, at least for now, to go with generalized additive models and perhaps some more like machine learning approaches, such as support vector machines. Okay, let's drink some water and jump into the results. All right. So first, a general overview of effort and observations. So in total, we have in this data set 81 helicopter surveys, but those are still ongoing, and 65 survey days in research cruises, which equals to 105 hours of helicopter surveys and 264 hours of research cruises. So congratulations to everyone who participated in this intense field work. I've represented the spatial distribution of this effort by year and by month. So effort started in 2016 with an Oceanus STEM cruise in September, but most of the effort is concentrated since February 2019 when the USCG surveys started. And it's been almost uninterrupted since then, except for the month of April and May 2020, obviously because of COVID, these surveys unfortunately couldn't take place. Now, although here I'm interested in large whales, these surveys are recording all marine mammal taxa. So in total, 15 different taxas were observed from aerial surveys and 21 in cruises. Although most of the time, individuals are not identified at species level. These surveys are also recording molars. It's really cool, molars, because these are indicators of leatherback turtle habitats. And finally, the helicopter surveys also record boats and crab fishing gear. Now, I mentioned a couple of parameters that were measured throughout the surveys to record detection and conditions, but the one that has been most consistently recorded across platforms is the buffered sea state. So on these plots, I'm representing the percent of effort time in each buffered sea state condition by cruise on the left and by helicopter transect to the right. So as you can see, there's quite a bit of variation in time and space here, with some pretty rough conditions, with buffered sea state going up to 6, 7 in some of these research cruises. But the conditions are quite consistent from one helicopter transect to the other. And most of the time, 75% of the time, the conditions were actually pretty good, like in buffered sea state lower than 3. However, wind is not the only thing affecting visibility. Obviously, fog and rain and other parameters are affecting detection. And we are now recording also a general qualitative index of sightability, something that goes from very good to very bad and just integrates all of these aspects together into a qualitative index that might sound a little simplistic, but is actually pretty integrative. Now, whale detections. So we had 574 baleen whale group observations, which if we convert it to individuals is equal to 655 humpback whales, 74 blue whales, 70 figure whales, 78 fin whales, and about 200 unidentified whales. I represented this number of individuals observed per transect in the helicopter surveys and per research cruise to the right. The proportion of species is quite consistent from one survey to the other or one transect to the other. The humpback whales are the most commonly observed species, but what varies a lot is the actual number of individuals per transect per survey. So if we look at the right, for example, here, in September 2020, there were more than 150 individuals observed, although in September 2018, so 2 years earlier, at the same time of the year, they only observed 25 individuals. And this is mostly related to big feeding whale aggregations. If the vessel happens to go for one of these just by chance, then the number of individuals goes up really quickly. Now, where were these whales observed? So here represented, mapped all of the observations from helicopter surveys to the left and from cruises to the right. And well, they're a little bit over the place, but there seem to be a few hotspots. So for instance, around Port Orford here, Newport, also in the helicopter surveys, Coos Bay, and in this canyon of Astoria. So I'm going to zoom on an area that I think will be of greatest interest to this audience. So these are all the helicopter observations and cruise observations between Florence and Lincoln City. And again, we can see that there's a couple of hotspots where there's more observations. So the Shelf Break here and here, the Ekata Bank and Stonewall Bank here. And generally, all of these observations are concentrated in waters deeper than 40 fathoms, except for gray whales. And I'll talk about this 40 fathoms line later on in this talk because it's a very important line for management. So we'll see the importance of this distribution later. Now, the distance of detection, first I compared it between species because some species that have are just larger, have higher blows, and have specific behaviors might be easier to see from far away. But this is not actually something that I detected so far in our data set. So I pulled all species together for the detection functions and only removed the unidentified whale species that had been observed at greater distances. So I excluded those for now. Now, the most important thing here was to compare platforms. So I represented the distance observed from the cruises at the top and the helicopter surveys at the bottom, and calculated the estimated trip width for these two platforms. And what we can see is that whales are actually detected at greater distances from research boats, about 2,100 meters from research boats and 867 meters from helicopters. So this is quite counterintuitive because given the height at which the helicopter is flying, we would have expected the observers to be able to see things very far away. But what we think is happening here is that the helicopter is passing by so fast that the observer is not able to detect things that are very far away, hence having this restricted estimated trip width. Now, of course, these detections are also likely to be affected by weather and sea states, but this is actually undetectable at this stage in the sample size as it is. But we're hoping that with more surveys coming in, we will be able to include that effect in the model as well. Okay, now this is a first insight, a first go at density surface modeling. So these are the models that we produce out of distance sampling data. So here I'm going to show you the results from an example, a generalized additive model that I've run with ROM's monthly average environmental conditions. And this model is focused on baleen whales, except for gray whales, because we considered that gray whales have very different habitat needs and need to be modeled separately from the others. So this model includes the blue whales, humpback whales, and fin whales. And it is relating the number of whales observed to a series of environmental variables and also to an offset. And that offset includes information about the survey effort, so the number of observers, the estimated trip width, and the speed at which the platform is traveling. Now, the topographic variables in this model are also included with the bi-season effect, so that the model would be flexible enough to detect a change in habitat selection from one season to the other. For instance, if whales were rather offshore at a certain season and came closer to the shore in another season. So the season here are split, well, the year is split in three. So there's January to April, May to August, and September to December. Okay, let's see the results. So I was actually happily surprised by the deviance explained by this model, which is 41.7. So it's really not doing too bad. And the significant effects in this model suggested that the distribution is mostly influenced by dynamic oceanographic features. So represented significance, the p-value stars with yellow circles here. And as you can see, bulk buoyancy frequency, isothermal layer depth, sea surface height, sea surface temperature, distance to shore are all significant. And from all of these, the one that had the highest contribution was the isothermal layer depth. So I'm going to focus a little bit on that effect. So this plot here is representing the predicted whale occurrence as a function of isothermal layer depth, which is in meters below surface. So it starts at 0 at the surface and goes down to 150 meters. And what this plot is suggesting is that whales have a preference for shallower ILD. And we think that shallower ILD could mean a more well-mixed water column that could arise from coastal upwelling. Now, ILD is known to have an effect on prey and predator distribution in the California current system. So I think this is quite consistent with what we see in the literature, and it's an interesting pattern for this first test of density surface modeling on this data set. Now, how does this look in terms of predictions? So using this model, I predicted whale occurrence at certain times over the Oregon coasts. So I've been focusing on the month of January when there is a lot of fishing going on, but very few whales in the area. The month of May, when a lot of whales have already arrived from migration, and fishing is at the late season, but it's still going on. And that's when there's the highest risk of overlap between whales and fishing. And September, when there's low fishing activity, but a lot of whales in the area. Now, these plots, these maps are showing a low predicted whale occurrence in blue and a high predicted whale occurrence in red. And there's a clear seasonal pattern here, with generally less whales in January as we expected, and more whales in May and September. There are a couple of hotspots from in these maps. So there's a high predicted occurrence around Coos Bay, around Newport and north of Astoria, and also in the area of Port Orford in September. Now, seasonal variability is not the only thing affecting the distribution. There are also some annual effects. This is what I tried to show here by making predictions for 2018, 2019, and 2020. And yeah, there's quite a bit of variation in the predicted patterns, although I haven't represented here the uncertainty associated with these predictions. And I'd like to emphasize the fact that there are areas of greater uncertainty. For instance, the offshore waters that are less well represented in the training data sets are waters where we need to be more cautious in our interpretations. So that's the case for May 2020, where we have this huge area predicted with high whale occurrence. I don't know how relevant this prediction is. And unfortunately, we won't be able to validate it with in situ data due to the absence of helicopter surveys at this time of the year due to COVID. But it's just important to emphasize the uncertainty in these models. Now, I'd like to draw your attention on an interesting pattern that was predicted by our models, suggesting that nearshore waters are less suitable to whale occurrence in late spring and summer. And this pattern nicely aligns with the new regulations accepted by the Oregon Department of Fish and Wildlife Commission that will impose a 20% reduction in crab pods and a prohibition on fishing at depth greater than 40 fathoms starting May 1st of this year. So on this map, the overlaid 40 fathom line with predicted whale occurrence shows a low probability of finding whales within the area where the late season fishing will be taking place this year. So I think this is suggesting that this new regulation might well be an effective way to reduce whale entanglement. Now, my job is not finished at all. There are many aspects of these models to question before reaching a robust conclusion. First, I'll be comparing the satellite and ROMS-derived environmental variables in terms of coverage and resolution to decide on which source is most appropriate to our project. I'll also be comparing temporal scales, looking at monthly variables, daily variables, and lagged variables. And finally, working with different approaches, modeling approaches, GEMMs, but also SVMs and potentially others, that other methods that might be more complex and integrates interactions between variables. Finally, I'll be splitting my data sets to conduct cross-validation and external evaluation of the whale distribution models. And this is a very, very essential step to limit overfitting and ensure that the model would work in different climate regimes and different years and so on. So between other things, I will be using opportunistic sightings from citizen scientists to perform this external evaluation of our model. One of the OPAL project objectives is to conduct outreach and promote a productive communication between citizens and scientists over the issue of whale entanglement. So Whale Alert, I'm showing here, is a mobile application that may be used by anyone out there who wishes to help record whale sightings within Oregon waters. And this analysis, this work is what these sightings will be used for, between other things. What now? So once we've produced a robust model that has been cross-validated and evaluated with external data sets, we'll be in the capacity to produce predictive maps of distribution, potentially in near real time, and then assess overlap with fisheries to see where and when co-occurrence occurs between whales and fisheries. And from these maps, we'll be able to derive maps of risk of entanglement in Oregon waters, which will provide information needed to make effective regulatory decisions. Okay, with that, I'd like to thank you for your attention. And I think I'll stop sharing my screen and read the questions from the chat box and try my best to answer those.
Great, thank you so much. As we're kind of transitioning here and you stop sharing your screen, folks, go ahead and put in any questions you might have in the chat box so that we can answer those. And you might see that there's one there right now for you that you can see.
All right. So the question is, what were the units on the vertical scale in that isothermal layer depth plot, I guess, the ILC? Sorry, these units, they did not show predicted occurrence actually, because it's a centered scale. So it's centered around the mean of the prediction, and that's why those units, I think, went from 0 to -1, -2, which obviously is not predicted whale occurrence. Sorry for not explaining that in more detail.
Great. As folks are adding their questions, I'm going to just ask, I'll wait on my question. There's another question that just popped up.
Okay, what is the spatial resolution of current predictions shown raster cell size? Yes, those predictions that I shown were at an 11 kilometer resolution, because that's the resolution of the ROMS environmental data. So I don't know if that was the question, but I agree that it's not as fine as we would want, and that's why satellite data might be of use as well, because some satellite products have a much higher resolution of 1 to 4 kilometers.
Yeah, I was kind of curious how the patchiness of the whales with the group that you looked at in comparison to the resolution that you were looking at. If you could talk to that a little bit, and maybe if you were expecting to see something different with the grays and their patchiness with the group that you displayed today.
Yeah, I think there definitely seemed to be some form of spatial autocorrelation in the data. If you see a whale, you have a great chance to see another whale next to it rather than nothing. And this is an aspect that these models will definitely have to account for, which is not the case yet, not in those preliminary models that I've shown. And yeah, I haven't done this work yet, but I'll be comparing different spatial resolutions to see which ones match the patchiness, the whale pattern best, and capture that fine scale habitat selection.
Thank you. You got a couple more questions that are coming.
Yeah. Wow. Okay. A lot of questions. Okay. How do your models compare to those put together by Elizabeth Becker et al. For the larger California currents? In other words, do the important environmental variables for your models match theirs? And are the relationships similar? If not, why do you think this is? Yeah, so it will be of great interest for sure to compare those maps with other efforts of well distribution modeling that have been done in the California currents, because these models are very preliminary. I haven't really compared them yet, but the relationships such as shallow ILD, and these models also show the preference for colder waters, high wind stress curls. These are parameters that have also influenced whales in the same way in other models that have been done at larger scale. So for now, I would be cautious because I haven't properly done this comparison, but it looks like the patterns match. But it will be an essential step because it's very interesting to see from different data sources and at different scales if the ecological relationships still hold. Any evidence of niche partitioning in the blue and humpback whales? Any inference about primary prey of the two species? So of course, this model for now is just pulling together blue, humpbacks, and thin. So we're not with this model in the capacity to look at niche partitioning between species. I'm hoping that by the end of the year and with a lot new surveys and observations, we'll have enough blue whale observation to be able to look at those differences. For now, the blue whale observation are perhaps a little limited to be able to look at this, but I will certainly model humpback whales separately from just the pooled baleen whales.
So the next question is from Beth. Do you see that one?
Oh yeah, this is okay. Maybe I'll look at this one first. So I think it's an answer to that question about the units on the vertical scale in the isothermal layer depth scale. So I believe that you said that the assumption is that detection is assumed to be 1 if whales are on the track line. I would think that with the helicopter that it might be considerably less, given that whales might remain underwater during the time that the helicopter is approaching and passing over a whale or group of whales depending on their behavior. Am I missing something? Yeah, so given the speed of the helicopter, the probability to miss something in a certain area is higher than if we were passing at a lower speed. I'm actually, so I could go back to the presentation, but if you remember those detection distance histograms, there was that kind of problem with not having an absolute detection at 0 distance from the track line, But I agree, it's a bit surprising, but this happened in the research cruises rather than the helicopter surveys. So that kind of effect, which I agree could totally be happening with helicopters, is not actually detectable in the data at this stage. The observers are actually in the capacity to see directly below the helicopter. And strangely enough, this is something we had in the NCC and STEM cruise. So this is also something that with finer detection functions we can account for, and I'll be trying to work on that. I hope that answers the question, but no, you're not missing something. I'm curious what additional data are collected on the Shimada. Could you use some of this data, perhaps net samples of plankton in your models? Yes, indeed. There is some in situ environmental data and prey data collected from the Shimada, and that would be a great addition to our models, although it will help our understanding of, well, selection of the habitats with respect to prey availability, but it's not necessarily what we need to produce those large-scale predicted maps of occurrence. And these maps are essential for management. So there's different ways of going with this, but including the in situ Shimada and prey data is definitely, well, what we would like to do in the future, for sure. Okay.
All right. I don't see any others coming in at this time.
Let me know if you'd like more detail, if my answers weren't good enough.
Yeah.
Okay.
Great. And I'm assuming that if folks have questions, they can either reach out to you or to Lee for more detail. And it sounds like this is some preliminary work, so we'll be excited to see how this continues to progress and see those results in a little while, which would be great. Lee, anything to add?
Nope, just want to tell Solène, great work. Yes, it's awesome to see these results coming together. Even though they're preliminary, it's pretty exciting. So yeah, thanks.
Yeah, it's amazing to be able to pull all those different pieces together and still be able to give us something that we can look at and have some predictive power. So it's amazing work. You're probably seeing it, but everybody's saying thank you, great job.
Yeah, I can see it. Thanks,
and
guys.
all the good things. Probably from names you know as well. So
yeah, I wish I could be doing this seminar in person at Hatfield, but
Someday
hopefully, yeah, that's what I was gonna say. Hopefully someday soon, when travel is easier and it's safe to do so. Fingers crossed. All right, for everybody online, thank you so much for joining us, and hopefully we'll see you next week. If you happen to be on spring break, be safe and have some good time away from work. For everybody else, again, still be safe, and we'll see you again next week. And again, thank you so much for joining us, and Lee, thanks for bringing her to us and keeping her connected to us. So thanks everybody, and we'll see you next week. Bye now.
Thanks.